And now we are going to start with the reactions that have to happen before the citrate cycle can start. So we're ending in glycolysis with pyruvate, and now we have to get that pyruvate to acetyl-CoA so it can enter the citrate cycle. And we know that pyruvate has to be transported from the cytosol, because that's where it's made, that's where glycolysis happens, to the mitochondria, and that's through pyruvate translocase. And so it's really going to, now that it's in the mitochondria, it's going to serve as our source of reducing power for the cell, right? It could serve really as reducing power for either the citrate cycle or gluconeogenesis. Just kind of depends. So when the energy charge is low, pyruvate is going to be metabolized by this multi-subunit complex. It's really pretty complicated, called pyruvate dehydrogenase. And so that's what this section is all about, pyruvate dehydrogenase. It's going to catalyze the oxidative decarboxylation, right? We're going to lose carbon in the form of carbon dioxide and turn pyruvate into acetyl-CoA. It's really a 5-step mechanism that is going to require 3 different enzymes. So we're going to look at the biochemistry of each of the reactions, and then what coenzymes are required by these 3 enzymes. So usually enzymes use their amino acid side chains to catalyze chemical reactions in their active sites, but really amino acids have a pretty limited chemistry that they can accomplish. And so cofactors and coenzymes can help give some more variety to what enzyme catalysis can do. So in redox reactions, they function as electron carriers, right? And so if you have dietary deficiencies of some of these coenzymes, then you're going to get some kind of metabolic disorder, right? And this is how it leads to their discovery. If you don't have one of these electron carriers that you should have gotten from your diet, then you're going to have whatever disorder. And that's how we identify vitamins. It's kind of cool. So our 3 enzymes that are going to happen are pyruvate dehydrogenase component. We call this E1. E1's a whole lot easier. Then you have dihydrolipoyl transacetylase, and that's E2. Then we have dihydrolipoyl dehydrogenase, which is E3. Okay, that's just kind of our common terms for how we do this. And so if we look at what actually has to happen, we're going to start with pyruvate. And I've just drawn pyruvate really simply. And the things that are going to be required to make this happen, right? We a lot of times say, as long as you say this reaction is catalyzed by pyruvate dehydrogenase, you don't have to list all of the required coenzymes, but typically in chemical reactions, you're going to see, okay, we need a CoA that's primed and ready. And so that's why we have this SH group here, because that SH group, the sulfur, we're going to lose the hydrogen, and then that's how we're going to bind coenzyme A to our acetyl group from pyruvate, is through that bioester bond. And so what else are we going to need? We're going to need NAD+, and then when this reaction has happened, we'll release a carbon dioxide, right? This is a decarboxylation reaction. We'll have NADH, and then we'll have our hydrogen ions. And so we're going to look at each of the coenzymes that are required in order to make this happen. But basically, we're going to get a decarboxylase for 2 carbon groups. We're going to transfer those 2 carbon groups until we get to acetyl-CoA substrate. So those 2 carbon groups that are going to eventually end up on acetyl-CoA are going to kind of get bounced around until we get to the end and we're ready for the citrate cycle. Okay, so if we have a low energy charge, we said we're going to go through citrate and oxidative phosphorylation. We're going to make ATP. If not, then our energy charge is high. We don't really need all this. And so we're going to skip citrate, and we're going to go and we're going to turn those acetyl units into things to store fatty acids. All right. So if we're looking at our very first cofactor here, coenzyme, that's coenzyme A. And so this one's derived from vitamin B5, and vitamin B5 is panthenoic acid. You don't have to know You don't have to know which vitamin is which. It's okay. But this one's going to be used to generate acetyl-CoA, right? So you have to have something that is going to carry this acetyl unit in an activated thioester bond. If it doesn't have a really high standard free energy of hydrolysis, it's not going to go through the citrate cycle. So that's the whole point, is to get this acetyl group into a high standard free energy for hydrolysis, and then it can go through our citrate cycle. So we need this ideal acyl carrier compound in order to make the whole reaction work. And so a lot of times what we do is we just don't show all of this stuff. We just say that's coenzyme A, and we say that there's a sulfhydryl, right, group right there. And so that's very reactive. And so that is going to participate in our reaction, and we're going to gain a group right here, a 2-carbon acyl group, and that's going to be attached to that sulfur. And so that little group that I circled is from pyruvate. So you do not have to draw the entire structure, but you really have to know why it's important. But having coenzyme A is essential for life. If you do not have coenzyme A, you will be in trouble. And you need about 5 grams a day, but it's really, really easy to get from a balanced diet. Like, it's super high in chicken and yogurt and avocados and all kinds of really interesting things, but you're going to get plenty of it for the most part. Okay. Thiamine pyrophosphate, so TPP. TPP was actually found because there were people who had a deficiency in it, and it was called beriberi disease. And these patients had anorexia, they had cardiovascular problems, they had neurological problems, and it was in populations that relied on white polished rice. And when you polish rice, you actually remove the bran, and the bran, the outside covering, right, contains the thiamine. So we saw it in those patients, and then you also see it in alcoholics, right? And so if you want to make sure you have enough TPP in your diet, you eat brown rice or you eat fortified grains and cereals, right? But you want the whole rice. You want with the bran on the outside, right? Okay. So this one is, again, required for our pyruvate dehydrogenase. And what's really cool is TPP is also found in an enzyme of the citrate cycle, and that's alpha-ketoglutarate dehydrogenase. So alpha-ketoglutarate dehydrogenase is structurally very, very similar to pyruvate dehydrogenase. And so this coenzyme is required for both of these two reactions to happen. So really our functional component, right, is a place where we can attach aldehydes. We can have an aldehyde transfer. And I'm going to show you this in the big reaction in just a minute. All right. Next, we have alpha lipoic acid. And a lot of people also call this lipoamide, right? And this provides a reactive disulfide that is going to facilitate your redox reactions. So this is within the enzyme's active site, and we make it in both plants and animals. And so this one is not derived from a vitamin because we can make it, right? And so we have our reactive sulfurs here, right? And so we're going to actually allow them to participate and reduce, and we'll see it in the whole pathway. It's a whole lot easier to see it in the whole pathway right here. Okay, so here's kind of our mechanism, right? In our very first reaction, see, they kind of numbered here. We're going to start and we're going to have pyruvate. And in the very first reaction, we're going to have the E1 subunit, the pyruvate dehydrogenase, that's going to bind to pyruvate. And it's going to catalyze a decarboxylase reaction. Decarboxylase meaning we're going to release CO2, and we're going to form what's called hydroethyl TPP. So we're basically attaching a 2-carbon unit to the TPP. All right. So now we're going to, this is happening, and this is happening in the E1, right? So we're kind of following this track, and we're really wanting to watch these red atoms here. So here are the atoms that came from pyruvate, right? We started as pyruvate, we lost these in the form of carbon dioxide. So we're not really going to track those anymore. What we're really tracking are these red ones. Okay, so we attached it to TPP. So now what's going to happen here, we're going to go up this way, and the hydroethyl of TPP of E1 is going to react with our disulfide in the lipoamide group, right? So we have our disulfide right here, and that is going to react with the TPP. And so what's going to happen is we're going to regenerate our TPP, right? So it's ready to go for the next time. And then at the same time, we're also going to make acetyl dihydrolipoamide. And here are 2 carbons, our acyl group that came from pyruvate here, and they are attached to the lipoamide through a thioester bond. Okay, so now we're going to try to hook this to coenzyme A. So the E2 acetyl dihydrolipoamide group is going to carry that acetyl group from the E1 catalytic site, and it's going to like swing around to a gap. There's kind of a big gap in the complex, and you don't really have to know about the structure, to the E2 catalytic site. So this is the point where we're going from E1 to the E2 catalytic site, and it's going to react with the coenzyme A, and we're going to generate acetyl-CoA. So that's great. This is the substrate that we need for the citrate cycle to go. So our citrate cycle is ready, but our enzyme has not been regenerated, right? So we need to make sure that we finish regenerating our enzyme. So the dihydrolipoamide group then swings over to the E3 subunit. So here's E3 is kind of in pink, the dihydrolipoyl dehydrogenase, where it's reoxidized, right? Because we need to regenerate our disulfide bond, and we don't have that yet. So we're going to regenerate our disulfide bond. And it's done through a transfer of 2 electrons and 2 protons to a disulfide on the E3 subunit, right? So we had this disulfide on the E3, and now we've lost the disulfide, but we've gained it on the lipoamide. All right, so then our E3 is actually linked to FAD. So FAD is going to get converted to FADH2 at the same time as we regenerate the disulfide on E3. But we're not quite done. The E3 FADH2 coenzyme is then reoxidized in another redox reaction. It's going to transfer 2 electrons from NAD plus to NADH. Well, to make NADH and H plus. And so now we have regenerated the entire enzyme. So you're not done with the pathway until the enzyme is ready to go again. And so this this enzyme, this pyruvate dehydrogenase, is going to determine if this pyruvate is going to go into the, how much of this pyruvate is going to go into the citrate cycle, right? So this is a really, really, really great regulatory step. There's a lot of different regulation that happens to pyruvate dehydrogenase. And we talked about the different ways that enzymes in general can be regulated, right? And how reaction speeds can be changed and things like that. So we know really that acetyl-CoA has 2 fates in the cell. Either you're going to go through the citrate cycle once we make it and get converted to ATP, or we have really high energy, and so we're going to send it off to be turned into fatty acids and or proteins, right? That depends on what we need. All right, but it's important to remember that this reaction is irreversible, right? All those reactions where we have lots of regulatory items around it, it is going to be irreversible. And that is because it has a very high negative delta G, not prime. I don't know why I put that extra one there. All right. And that is negative 33.4 kilojoules per mole. So very, very irreversible. So so this enzyme is super, super important in animals that lack enzymes to convert fats to carbs, right? So that's us. So then we can't reuse that acetyl-CoA for glucose production when our carb levels are low. So it's really, really important for us to be able to control this. Oh, look, and delta G was right there. All right. So this is really kind of cool mechanism. So you know that arsenic was used in the past to kill off kings and queens, right? And if you had arsenic poisoning, you had ulcers on your skin, all kind of lesions, and you had a high risk for cancer, a high risk for death, right? And they think this was possibly what was used to kill Napoleon Bonaparte. Little history lesson there. But more recently, it was in contaminated drinking water in Bangladesh in the 1990s, and it was leached in from the soil. And so the problem with arsenic is that it prevents the pyruvate dehydrogenase from regenerating. Remember how I said this was really, really important? And so what it actually does is, here's our active form. When you ingest it, it turns into arsenite. And what it's going to do is it's going to bond to these SH groups. And then instead of being able to regenerate them into a disulfide, now you've basically irreversibly bound to them, and the enzyme can't be regenerated, so you can't use it. So you can't make any more acetyl-CoA. It's very, very dangerous. All right, so let's kind of go over our regulation of pyruvate dehydrogenase. So allostery is really, really important. So small molecule binding. And we know that it has a binding site for NAD+, but what's really cool is that NADH competes for exactly that same binding site on E3. So this is negative regulation. If you have lots of NADH, then you're going to inhibit pyruvate dehydrogenase. And this is going to occur if oxidative phosphorylation later on down the road slows down, right? Because if it slows down, you don't need to give the electron transport cell more substrate. So usually oxidative phosphorylation is going to convert NADH to NAD+ in the mitochondria, right? We've talked about that happening in the cytosol, but this is actually in the mitochondria. And then immediate feedback inhibition is through high levels of acetyl-CoA, which competes for coenzyme A binding to E2. So that's a negative regulation. And you just want to immediately, if you're not using up the acetyl-CoA that you're making, you don't want to make any more. And then you have covalent modification through phosphorylation. So if you phosphorylate serine residues on pyruvate dehydrogenase now, if you add the phosphate group through pyruvate dehydrogenase kinase, you're going to inhibit the catalytic activity. So this one is add the phosphate and inhibit, right? And then the pyruvate dehydrogenase phosphatase is going to remove the phosphate and it is going to activate, right? So this just gives us a covalent way to modify the enzyme's activity, which we've talked about many, many times before. So let's kind of summarize our different ways that we can inhibit and ways that we can activate the pathway. So we can inhibit it with high levels of NADH, right? Saying oxidative phosphorylation is getting backed up. Immediately through acetyl-CoA. If we have high levels of ATP, that's going to inhibit this cycle. And then we talked about phosphorylation, right? And that phosphorylation is actually on E2, but you don't really have to know that. All right, then our activating, kind of think about the opposites. So if NADH inhibits, then NAD+ activates. If acetyl-CoA inhibits, then coenzyme A activates. If ATP inhibits, then ADP activates. And the very last one that we really haven't talked about is calcium levels. So calcium levels activate our pyruvate dehydrogenase phosphatase. So these activate the phosphatase. And the phosphatase dephosphorylates, which activates, right? And this is really, really important, mainly in muscle cells, because calcium levels regulate muscle contraction, which is going to tell you how much energy the cell needs. Okay, so remember, we're going to convert from pyruvate to acetyl-CoA with pyruvate dehydrogenase. We have 3 protein subunits that you need to know. There are 5 cofactors, right? And those cofactors A lot of them, but not all of them, are enzymes because we've lost the ability to make them over time. So we have to consume them. You need to know your 3 reactions, right? E1's a decarboxylase, right? So we transfer that, that we cleave off one of the carbons, right, and release it as CO2. Then we send the other carbons through to the hydroxyethyl TPP. E2 is our transfer of 2 carbon groups to the lipoamide, right, through acetyl dihydrolipoamide. And then E3, the acetyl group, is passed to coenzyme E. All right, so that is a good summary. And then next, we'll talk about the actual enzymatic reactions of the citrate cycle.